Animal behavior is hugely influenced by chemicals, and one such chemical is a hormone. And we have a whole lot of hormones in our bodies, and they exert different types of effects. That's what we're going to be looking at in this segment. So a hormone is an internal chemical messenger. It's actually secreted from glands in the body, and the target site are other areas of the body, other glands or areas of the brain. And there are basically two types of effects that hormones can exert, two types of influences that affect structure and function of an organism. These are not necessarily mutually exclusive, but they can be conveniently divided, at least for the purpose of talking about these things, as activational effects, which basically refers to the current hormonal state of the organism and how that ends up affecting behavior. Well, the second type of influence is organizational effect. These are earlier hormonal influences. In other words, hormones that act early in life, even before birth or hatching, but you don't see the behavioral effect until much later on. So, again, we're dealing with relative time scales here. That's why I'm a little bit hesitant to talk about these as really discrete. But again, for the purpose of convenience, we'll just look at them in terms of how they work kind of separately from one another. So let's start with activational hormonal influences. So metabolism, for example, is greatly influenced by a hormone called thyroxine that comes from the thyroid gland. It increases metabolism, increases heart rate, respiration, activity levels, behavior. So basically, overall effect on metabolism. If you don't have enough thyroxine, by the way, you'll have decreases in some of these processes as well. But under normal circumstances, you'll have secretion of thyroxine from the thyroid gland that causes these various physiological and behavioral effects to increase. Beak color. Okay, that's influenced by levels of the sex hormone testosterone, the male sex hormone testosterone. So here we have two males, and you can see that the one over on the left has a redder beak, the one over on the right has a paler beak, and the redness is influenced by higher levels of the sex hormone testosterone. Here is a newborn chicken, and so testosterone affects that as well. For example, the chick over on the right is a normal 3-day-old chick, the way they would normally look. If you inject a chick with testosterone, within a day or two, it's going to grow a comb on the top of the head and wattles underneath the beak, and that's what we see over on the left. So testosterone basically is inducing sexual characteristics. That's its normal role. And normally you wouldn't see that in a newborn chick, but again, you can see how if you actually inject it, you'll have this activational influence. And in fact, they'll even attempt to engage in courtship-type behavior like crowing when you inject testosterone. So there's our testosterone-injected chick. It's actually crowing like an adult would crow, an adult male, and it sounds something like this. Much higher pitched because it's a much smaller bird. Adrenaline from the adrenal glands. Okay, that has an important activational effect on memory.
This rat at the University of California, Irvine, is about to get a better memory thanks to Jim McGaugh. There's food at the end of 4 of these arms. Entrances to each of the other 4 are blocked by a plastic window. Once the rat has eaten the available food, the windows are removed and food is placed in the previously blocked channels. After several trials, the rat learns to enter only the newly opened arms, ignoring the old ones it had already cleaned out. "18 hours later, however, the rat has forgotten the secret and checks out the old arms as well as the new. But this rat is getting some help. A shot of adrenaline immediately after learning the task. This time, after an 18-hour absence, his memory of the maze was as good as new.
He remembered where he had been before and only went to the arms that he hadn't been to. So he performed the task very well, perfectly, 100% performance.
Adrenaline is the hormone responsible for the fight or flight response, the surge of energy we and other animals get when we're threatened. Jim McGaw's experience with rats suggests the adrenaline rush is doing more than allowing us to run fast.
It also would be a very good idea to be able to remember where the predator was and what happened so that the next time that animal can avoid that situation or to minimize the probability of being eaten the next time. So the same hormones which were involved in generating the fight or flight response, we now have discovered work on the brain so as to make stronger memories.
So what would happen in a stressful situation, which for rats means having to swim, if somehow adrenaline is removed from the picture? This rat is trying to find a transparent underwater platform. Eventually, he has to be shown where it is. Three days later, and he finds the platform quickly. Like his colleague, this rat has also been shown the platform. But moments later, he gets an injection of a drug, a beta blocker that blocks the effects of adrenaline. When this rat's tested 3 days later, it's as if he's never been here before. So for rats, adrenaline seems central to making stronger memories.
Okay, let's look at some of these organizational influences of hormones. So again, the organizational effects typically occur earlier in life, but you don't see the effect of that until a much later time, unlike the activational effects where you get a more or less immediate effect. So there is this area of the brain, it's a nucleus, a group of cell bodies. It's called the sexually dimorphic nucleus of the medial preoptic area. That's a mouthful. But it's involved in sexual behavior. So, for example, it influences male mounting behavior and female lordosis. And you can see these various postures in these photographs. Obviously, the male's on top in the white rat, and the female is exhibiting lordosis, which is a posture that she adopts indicating that she's receptive to that particular male, and then copulation will immediately ensue, as you see here. So, the sexually dimorphic nucleus of the medial preoptic area is involved in this sort of behavior, but it's set up prenatally. The sexual dimorphism of the nucleus occurs prenatally, and there it is in red, that area that's circled in red right there. That's where you'll find the MPI, the medial preoptic area. And here it is stained in these three slides as well. So what you're looking at over on the left is the nucleus in a male, and in the middle, the nucleus of a female. And you can see why it's sexually dimorphic. It's much larger in the male than it is in the female. And again, that's something that occurs prenatally. And by the way, if you were to inject a female with testosterone, the male sex hormone, then of course it'll enlarge like we see over on the right. But of course that normally would not be the case. So why is this an organizational effect? Well, I think I've already said it, but basically the dimorphism happens prenatally because of prenatal androgen, and you're not going to see the sexual behavior occur until the animals have reached puberty at a later age. So the hormonal effect happens very early, the resulting behavior much later. Oxytocin and vasopressin affects bonding in voles. So we have these two very similar looking voles. The prairie vole has a lot of receptors for oxytocin. By the way, the difference between oxytocin and vasopressin: oxytocin is the hormone involved in females. Vasopressin is the hormone involved in males, but they work very, very similarly. They're just different hormones in the two different sexes. So the prairie vole has a lot of receptors for oxytocin in the case of a female, or vasopressin in the case of a male, and they're very monogamous. They have very strong parental bonds. However, the montane vole has fewer receptors. They are very promiscuous and very weak in terms of parental bonds. So here are two videos that describe this situation.
These are prairie voles, rodents that live in family units. The breeding pair is strictly monogamous. Prairie voles mate for life. On the other hand, their cousins, the montane voles, play the field. They are quite promiscuous critters. Commitment means nothing to them. Although these two species look the same, their brains differ in one significant way. The areas in green contain receptors for oxytocin. The green oxytocin receptors of the family-oriented vole are shown on the left. On the right, the less committed vole just can't compete. Distinct differences become apparent in the behavior of the two species when newly born young are separated from their mothers. The montane vole, with few oxytocin receptors, is indifferent to her young, nor do the babies feel a very strong attachment to their mother. Then, the prairie vole is taken away from her young. Already, the bond is obvious. When placed back in the cage, the prairie vole quickly runs to her young to cover and protect them. They cling to her just as fiercely.
So what matters here, of course, are these receptors, not so much the level of oxytocin and vasopressin. And the receptors are set up prenatally during early brain development, but of course parental behavior is going to kick in much later. And in fact, before you can have parental behavior, you have to have sexual behavior. And so that's what this video is describing.
Prairie voles are among the most doting mammals on Earth. Like some humans, males and female prairie voles form lifelong bonds, share parenting duties, and males jealously guard females. In contrast, a nearly identical cousin has adopted a more swinging lifestyle. When the promiscuous cousins have sex, there are no strings attached and no cuddling afterwards. In fact, if they get too close, they're more apt to fight than to playfully nibble. What's driving these two distinct approaches to mating? And could the answer help us understand human relationships? When Young peered into the vole brains, he discovered that both species were releasing the same love cocktail as humans. But there was one big difference between the vole species. Only the monogamous voles had sufficient brain hardware in the form of receptors that could soak up the bonding chemicals.
Hormones like oxytocin or vasopressin can only act by binding to a receptor. It's sort of like a key in a lock. Well, when we looked in the brain of the prairie voles, we found that these reward areas were loaded with oxytocin and vasopressin receptors.
The promiscuous voles have fewer receptors, and Young suspects that could explain their free-loving lifestyle. To investigate his theory, Young turned to a virus. His plan was simple. He injected a virus into the brain of a promiscuous vole. The virus sliced into the vole's DNA and inserted the instructions to create more receptors. Brain scan images revealed success. But would the new receptors change behavior? To find out, Young mated the virally altered vole with a female. And then separated the couple. Next, he tethered the mated female in a box. At the other end of the box, he tied down a temptress. And finally, he introduced the virus-injected vole. Would the male remember the days in the love nest? Or would he be back on the prowl? In an evolutionary flip-flop, the virally altered vole returned to dote on his mate.
So yes, the receptors matter a lot. Sexual differentiation is something that occurs prenatally. And of course, sexual behavior occurs much later. So again, this would be an organizational effect. So by sexual differentiation, we basically are referring to how you actually become a male or a female of whatever species you happen to be.
These are historic pictures of a living human fetus, the size of a button at 7 weeks. Images so crystal clear you can see a tiny pinpoint of pigment now coalescing into an eye's color. And soon a genetically timed hormone will carve this speck of tissue between the legs into a sex organ. A male penis or a female clitoris. But now, at 7 weeks, boy and girl bodies are the same. 9 weeks, startling changes. Lids now seal the light-sensitive eyes until birth. A recognizable face emerges. Brow, nose, chin. Even a working mouth that opens for our camera to flaunt a rudimentary tongue. As a nipple buds over the rib cage, the sex tissue elongates as well. The nub looks like a penis, but it could still become a clitoris. For at 9 weeks, the size of both are identical. The opposite sexes still the same. That's about to change. Dramatically. 15 weeks. Beneath the transparent skin, a filigree of vessels sends sex hormones to the brainstem. And over the full 1-inch length of the body. This fetus is becoming a boy. The sex nub, now a penis, fully fledged, and at 100 days, capable of a physical erection.
So what hormones are involved in this process of sexual differentiation? Well, it depends if you're a mammal or a bird, because they're different. So the so-called default sex, or the undifferentiated sex, the one that is not yet male or female, in mammals, basically, again, the undifferentiated sex would be female, but the prenatal action of androgen will masculinize the developing fetus, and so that is how you end up differentiating into a male if you are a mammal. So it's the prenatal action of androgen that masculinizes the otherwise female undifferentiated sex. In birds, it's the opposite. The undifferentiated sex is male. So the only way you're going to end up with a female bird is because of the prenatal action of estrogen, which will then feminize the developing fetus, and then you'll have a female bird. So that's how prenatal sexual differentiation actually occurs, through the action of these hormones. And then, of course, you'll end up with sexual behavior later in life. But this is at least how you get the two sexes in the first place. Now you can see this is where it gets a little bit wishy-washy in terms of, is this organizational or activational? I mean, it's activational, really, in the sense that once you start to get the prenatal secretion of androgen in the male or estrogen in the female, then you start to see the two sexes begin to develop, a male in a mammal or a female in a bird. That's kind of activational. But in terms of what will occur sexually, in terms of sexual behavior later on, that will occur much later. So yeah, it's again a shade of gray, which is why not only me but others are a little uncomfortable with totally separating the two categories of activational and organizational effects. So just, a little bit of a caveat there. Here we have a womb of a rat. So we have a whole bunch of developing rat fetuses, and you can even see the little rat paws right there. There's some right there, rat feet. There's a tail right there. So these are still in very early stages of embryonic development. Okay, so here is what can sometimes happen. Now, I'm going to just show you the direction of blood flow because it matters. So the way this is drawn, the blood is going to be flowing in that direction, meaning it's going to, we'll just look at one side, you'll have the same effect on the other side, in and out of that developing fetus, and then in and out of that one, in and out of that one, right on up to the top. Okay, so that is a genetic female, and next to it is a genetic male. So I've color-coded male and female. Females are red, males are blue. Okay, that's the genetic sex. Okay. In terms of what they'll actually end up being anatomically, could be affected by the prenatal levels of androgen. We're dealing with mammals here, so this is androgen. So this female might, there's no way of knowing for sure, but might become somewhat masculinized. Unlikely she's going to be a total change into a male. So what's going to happen is that blood is going in and out of this female, but then it's going to go in and out of this genetic male. It may pick up just enough extra androgen such that when it goes into this female, it somewhat masculinizes her. And then, well, there's nothing but males up there. Now, the one over on this side, even though she's next to a male, she will not become masculinized. I mean, there's no way, because there's nothing but females here, so she's not picking up any androgen from these genetic females. So one way to tell if there's been any prenatal masculinization of a genetic female that really is, in all other regards, female, is by just simply looking at the anal genital distance, because it's sexually dimorphic in male and female rats, and it's, as you can see, a much greater distance in the male and a shorter distance in the female. So if there has been some masculinization, it might show up there, and the female might show more of a male pattern in terms of anal genital distance. You might also see more rough and tumble play with other rats, things like that. We see it in humans, by the way, sometimes as well, with females that are somewhat tomboyish, may have been affected by some higher level of androgen in mom's blood. And so again, you can see those differences there in anal genital distance.
So we can actually reverse the behavioral, genetically determined sex roles, male-female, simply by manipulating the hormone environment during critical periods of development.
The white rat is genetically female. The black and white rat is male. This is normal behavior. These are two genetically female rats. One's been treated with male hormone and behaves like a male. This is a masculinized female paired with a male that was castrated and treated with female hormones. The roles are reversed.
Okay, prenatal masculinization. So there's our cute little rats right there. So those are activational and organizational effects of hormones.